This research was presented in the wood identification session at the 2023 Forest Products Society International Conference. Have you ever wondered how scientists determine wood species? Well, conventional methods rely on visual features using macro and microscopic analysis, and although we still employ them, there are some limitations. I'm Adriana Costa from the Wood Identification team at Mississippi State University. Today, I bring you an exciting advancement in DNA-based wood identification. In this presentation, I will introduce you to this preliminary research to detect Norway spruce lumber in the field using DNA. So these are my co-authors in this project: Frank Owens, also from Mississippi State University, Grant Morton from University of Wisconsin, and my colleagues from the USDA Forest Products Laboratory in Madison, Wisconsin, Grant Kirker, Amy Bishop, and Alex Wiedenhoft. But first, let me give you a brief historical overview on Norway spruce in the US. The first records of Norway spruce plantation in the US date back to the 1860s, when European immigrants planted Norway spruce in Massachusetts. And this was a super successful plantation because of the high survival rates and relative fast growth compared to native softwoods. Pretty soon there were around 58 plantations spread throughout Massachusetts, Vermont, and New York. In this picture, two unidentified men walking between rows of Norway spruce that were planted in 1882 and were 45 years old when this photography was taken. Norway spruce seedlings became a critical component of President Roosevelt's Back to Work program in the 1930s. But the Civilian Conservation Corps replanted thousands of acres of abandoned agricultural lands in the Northeast that were created by the Great Depression. And the purpose of the reforestation was to stabilize barren soils and reduce erosion. More than 113 million Norway spruce seedlings were distributed by state nurseries for these replanting projects in New England, New York, and Pennsylvania. And millions more were made available to the Corps for hundreds of projects that followed for the next 10 years in that region. So fast forward to 2016, the American Lumber Standard Committee approved US-grown Norway spruce into its spruce pine fir species group, SPFS, which stands for South. This in contrasting to the SPF that comes from Canada. SPF from Canada has its own mix of spruces, pines, and fir species, and there are differences in strength values between SPFS and SPF. The Canadian SPF has higher design values for most grades. Canadian SPF does not contain Norway spruce, and as I mentioned before, has higher mechanical properties for most grades. In general, the problem is that Norway spruce has these lower mechanical properties, and we cannot tell them apart. Anatomically speaking, all we can see in spruces is that they have these small and sparse resin canals and this gradual transition from early wood to late wood, and that's all. So this one on the left is a Norway spruce, and this one on the right is a North American spruce. This one specifically is Picea mariana. So it is important to ensure that imported SPF does not inadvertently contain Picea abies, which is Norway spruce, to prevent potential in-service failures. And adulteration is a hot topic that Dr. Owens will talk in his presentation, and I will link his video here. So how to reliably separate Norway spruce from other North American spruce species? Our solution was to develop a DNA method to separate Norway spruce from the North American spruce species: Picea rubens, Picea mariana, Picea glauca, and Picea engelmani, and we want to do that in the field. So this is just an overview of the protocol. Step 1 is probe and primer design. This step occurs in the computer. Step 2 is the protocol optimization that involves many tests. For what we need a lot of DNA, and because of that we extract, in this first step we extract it from leaves. Step 3 is the protocol validation, in which we actually use wood sections. So let's look at each step in more detail. So this is step 1, and we go to GeneBank, which is a database for DNA sequences, and we pull the DNA sequences from North American spruces and Norway spruce. This is not the total sequence. This is just part of the sequence that I'm showing here. So there are different softwares that we can use to align the sequences, which means that we want to match here the majority of these letters in the same sequence. Then we visually found these areas where the letters are the same for all North American spruces, but they differ for Norway spruce. For example, in this column here, we have G's for all North American spruces, but then A's for Norway spruce. There are other areas where they differ, but this area is where we found the more differences. So this is the region that we are going to target. So these are the regions that we are going to target using probes. So these are little fragments of DNA. The probes have a fluorescent reporter, and because we have 3 channels in our equipment, we label them as red for Norway spruce, yellow for North American spruces, and green for any double-stranded DNA. And our primers were designed based on the probe attributes. So that's step 2, the step that we start in the lab. And because we need to do many tests, we extract DNA from leaves, and because it's much more abundant. Then we need to do a fine adjustment on the best temperature and concentration for both primers and probes. And after we have everything figured out, we mix everything together in a tube. We add one of the species DNA there, and we run into this machine that is a qPCR machine that we call the toaster. So let me explain. This is the double-stranded DNA. In this stage here, the DNA is denatured, so we raise the temperature so the 2 strands come apart from each other. And this is the probe. So the probe binds, and this red circle here is the fluorescent reporter, and this is the quencher. This gray circle is the quencher. The proximity of the fluorescent reporter and the quencher will ensure that fluorescence will not be emitted at this point. So the probe binds to that target region that we identified in the first step, right? Then the primer binds at the region that it has to bind, and this enzyme, which is the polymerase, binds and starts the synthesis of DNA. So it moves in this direction, and then when it goes through the probe, it releases that fluorescent reporter. And because the fluorescent reporter is no longer close to the quencher, it emits a fluorescence that can be detected by the equipment. So everything working fine, we will validate the protocol in wood. We will cut these thin tangential sections of wood and add it into a tube containing a buffer that will help release the DNA into the liquid. And then we will add our mix of primers and probes, and then we expect to have results like this. When we detect Norway spruce, we will have green for double-stranded DNA. So green we will always have, and then we will have a red that detected Norway spruce. When it's an American spruce, we expect to have green for double-stranded DNA and yellow for North American spruce. All right, so in this preliminary research, we concluded that running a qPCR in less than 1 hour and with few steps, we can separate Norway spruce from North American spruces in the field with minimal training. And this would allow lumber inspectors to detect the presence of Norway spruce in imported SPF materials. Well, this DNA wood identification method provides a valuable tool to ensure the accurate identification of wood species, particularly in this study, Norway spruce within the SPF species group. And by implementing this method, we can prevent adulteration in imported SPF, avoiding potential failures, in-service failures, and also we can promote the integrity of wood products in the market. Thank you for joining us. If you have any questions or comments, please feel free to share them below. Stay tuned for more exciting developments from the wood identification team at Mississippi State University.